Biochemical energy. If anything today, I hope you walk away appreciating how amazing your body is. We're going to learn a little bit about it. So the whole gist of chapter 21 is for you to understand how your body creates energy, and it's the energy that sustains everything the body does, okay? So this is going to be a walkthrough of how our body takes what we feed it, the air we breathe, all of that, and it's combined together in these really complex pathways, but it's really cool at the same time to make ATP, and how we use ATP to power all the reactions that our body has to do. So biochemical energy, that's why it's titled this. Now, the major things we're going to be looking at. So here's the Krebs cycle. Anybody just, you can raise your hand, nod your head. Y'all have heard of the Krebs cycle. Has anybody actually studied it before? Like the steps involved? Okay. So we're going to get to that officially tomorrow. We're going to stop just before that. And then we're going to really assess ATP. We're going to assess some of the things about ATP today. And then tomorrow we'll get to oxidative phosphorylation, the electron transport chain, right after we talk about the Krebs cycle, because they're tied together. They feed in one to another. But we are going to really today assess kind of the strategies that our body uses for these things to happen. So we'll really get to these tomorrow, but we're going to assess a lot of kind of the foundational background work to set us up for those today. All right, so I'm going to give you an example. I think this is a great example. Anybody know what this is a picture of? Looks complex.
I mean, isn't it like a dam that like generates energy?
A hydroelectric dam. Anybody can tell what part of the world we're in? These things on the top, give it away at all? We're in China. Okay, so this is the world's largest hydroelectric dam. We have some big ones, the Hoover Dam, you may have heard of it in the United States. We've got, a lot of large hydroelectric dams. But this is the world's largest. So China just thinks they have to be the largest at everything. They have the largest wall and the largest dam, okay? But this is actually a good example for how our body makes ATP. And here's why. I'm gonna zoom in on this picture. So this is kind of a small little graphic for how hydroelectric dams work. You have the dam, okay? And what happens is the water is trapped. I'm gonna use red, I think you can see red better. The water is trapped behind the dam and it's held there. Well, it is then navigated through the dam, it passes through the dam, and the downward motion of the water, okay, using gravity, pushes this turbine. Okay, so it's called mechanical rotation. It rotates this turbine. Now the turbine is then used to run a generator. So the mechanical turning powers the generator, and the generator produces electricity. Okay, so that's in simplest form how hydroelectric dam works. How does this apply to ATP? Okay, so when we get to oxidative phosphorylation tomorrow, I want you to remember, the dam is going to become a membrane. Okay. The water behind the dam is going to be a stockpile of hydrogen ions. Okay, so in a controlled manner, the hydrogen ions are going to run through the membrane, and they are actually going to rotate and turn instead of a turbine, a protein. All right, so literally this thing rotates and it turns just like a turbine. And as that turns, instead of electricity, we're going to produce ATP on the other side. Okay, so maybe that will help you, maybe that will stick in simplest form. But we are today going to check off metabolic strategies, pathways, and then tomorrow we'll get citric acid cycle, oxidative phosphorylation. Okay, so I have to review a little bit before we get into this, because a lot of this builds on one of the most complex chapters from the preceding course, 1402. So anybody seen this equation before? Delta G equals delta H minus T delta S. Hopefully you've seen it before, but if not, I'm going to remind you. All chemical reactions have an associated energy value. Some chemical reactions are favorable, or we'll use the word spontaneous. The thermodynamics word is they're exergonic. Some reactions are not spontaneous and they're not favorable. but they still have to happen. And we call those endergonic, okay? And that's what this equation is telling us. This value, delta G, is known as Gibbs free energy. You might want to write that down. Okay, and we can see delta G depends on delta H and T and delta S. Anybody remember what these terms are? Delta H, delta S, T?
I know delta means like the change of.
Yes.
I think the T is temperature.
T is temperature. Abby snagged the easiest one.
Yep, you know, taking the easy way out.
Anybody remember delta H? Who's like Googling it really quick now? Okay, this is enthalpy, change in enthalpy. Delta S is entropy. Okay, entropy is like a measure of disorder in the system. Now, I'm not really going to get into delta H and delta S here. I just want you to know delta G depends on these terms and these variables, all right? So every equation, if you add these things up, if you plug in, if you have a value for delta H and a temperature and a value for delta S, you can plug it in and you can calculate delta G according to this equation. So a negative delta G, we can look at the sign of delta G. This is favorable. Okay, or you might hear me say spontaneous. The chemistry word for it that I mentioned earlier is exergonic. Right? Ultimately, what this means is the products are more stable when you're done than the reactants. And that's favorable because we like to make things that are stable. Okay, so what that would look like, if anybody remembers energy diagrams, the y-axis here is free energy. And the x-axis is as the reaction proceeds, reaction progress. Okay, so every reactant that's used in any chemical equation has some associated energy to it. Let's just say our reactants have this much energy, this much free energy. Okay, then you did see me draw this with activation energy. I forget if it was yesterday or the day before, but we've got some sort of energy barrier that we have to clear. Okay, the activation energy. And then for an exergonic reaction, we have this downhill slope, and the bottom line is the energy level associated with P for the products, okay? Because we know all reactions are some kind of reactants and they make some kind of products, okay? So this would be exergonic. Why is it exergonic? Okay, because if you follow the change on the y-axis, ultimately that change is downward, negative delta G, okay? A negative change in free energy between your reactants and your products. Does that make sense? If you mark off the level between the reactants and the products, this change is downward, it's negative. So that's favorable. Okay, but some reactions have a positive delta G. This is unfavorable. Okay, or you might hear me say non-spontaneous. I'm going to abbreviate. And we call this endergonic. Okay, so here's the thing. Delta G is a great way to measure favorability or spontaneity chemical reactions because it's kind of the sum of everything that it equals. Instead of separately measuring the delta H and the delta S, which has to be done, but if we have a delta G value, we can just look at the sign and say, oh, negative, favorable, oh, positive, not favorable, okay? So that's why it's kind of nice, because it's a simplified term. It's the sum of everything that it equals. You just have to look at one thing with delta G. An endergonic reaction, I'm going to do a different color, might look like this. If we start with the same energy level of our reactants, maybe we also have the same energy barrier, but our products are higher in energy. So this is not a huge change in energy, but you can see if we mark off the difference between the energy level of the reactants and the products, ultimately the change in free energy is upwards. So that's why we say overall this would have a positive delta G, because you're moving up on the y-axis as you go from reactants to products, okay? So all that to say, delta G is really important. Every single chemical reaction can have a calculator associated delta G value, whether we know what it is or whether we need to calculate it. One really good example is the oxidation of glucose, okay? Some people might say the combustion of glucose, because look what you're doing. You're reacting glucose with oxygen, you're getting CO2 and water. We know the production of CO2 and water is the hallmark of a combustion reaction. But ultimately what we're doing is we're oxidizing. glucose, okay? Now look at the associated energy value. The delta G, it's kind of crammed together, but the delta G here is a negative 686 kcal of energy per mole. So remember, units of energy are kilojoules or kilocalories. So that's why it also says or kilojoules, okay? I'm not going to have you convert between kilocalories and kilojoules on the exam, okay? But I may give you a delta G and want you to interpret some information from that, all right? So oxidation reactions tend to be favorable in the body and in nature, okay? Write that down. Oxidation reactions tend to be favorable, which means that the opposite of that, reduction reactions, may tend to be endergonic or not favorable, non-spontaneous. Because any reaction run in reverse is going to have the opposite, same value, just opposite sign of delta G. I'm going to say that again. Any reaction run in reverse is going to have the same magnitude but opposite sign of delta G, and that's what this figure right here is showing you. So, by the way, this is a little bit confusing because they flipped the products and the reactants. So you have glucose here, but you have it right there on the side. You have oxygen on the reactant side. It looks like you have it on the product side here, but it's the same reaction. It's just flipped. Okay, so orient yourself with this figure. Here's our delta G for oxidation of glucose to make CO2 and water. We just saw it's a negative 686 kcal. So the opposite of the oxidation of glucose, which is what our bodies do, the opposite of that is photosynthesis, and this is what plants do. So it's really kind of nice. Plants do the opposite, and then we take what plants make and we break it down and get what we need. Okay. So plants take CO2 and water, they run photosynthesis, and they make glucose and oxygen. And then we eat plants, we get the glucose and the oxygen. We breathe oxygen too, but we take those things and we oxidize them to get energy. Okay, because the oxidation of glucose is favorable, releases a lot of energy. The photosynthesis, though, the formation of glucose has a positive delta G, same value, different sign. Okay, so is everybody tracking with me as far as what the signs of delta G communicate to us? Okay, if y'all can join us with video, I know that some people have valid excuses, hot spots and things like that, but if you can, please do.
I'm here.
Okay, I know. I just like to see y'all.
I'm just like unbelievably close to my phone.
So all we would see is like Cameron. Whoa, about fell out of my chair. All we would see is your nose. Is that what you're saying?
In my forehead, yeah.
Okay, fair enough, Cameron. All
Sorry,
right.
my dogs keep tearing stuff up, so I have to keep getting up and picking them up and chasing them and pulling things out of their mouths. And I just
I
didn't want to
understand.
distract everyone with my camera on. I'm sorry.
Okay, you bet. I get it, guys. Thank you for letting me know. Okay, so the last thing I'm going to finish with is that living systems take advantage of how these things work in concert. All right, living system use pathways. We have to do both things. Sometimes we have a lot of reactions that are just run in the favorable direction, but we also have to perform reactions that are unfavorable to the body. Okay, example. Washing dishes is not a favorable activity for me, but I have to do it anyways, right? Studying for your chemistry exam Monday, not a favorable activity for you, but it's got to get done anyways, right? So there are times the body has to do both, okay? And we have both kinds of pathways. So how do we power these things? How do we make them happen even if the sign of delta G is not favorable and that reaction is not favorable to the body? It requires an input of energy versus releasing energy. Well, this is where mitochondria come into play. They are the powerhouse of, they're kind of like the energy plant. So if y'all think about anybody drive, I use this example sometimes in my class, past Russellville, there's a big nuclear power plant in Russellville. Sometimes you can see like the smokestack from the interstate. But that provides so much energy. Hydroelectric dams provide so much energy for the surrounding area. That's what mitochondria do for our cells. So I'm going to zoom in on this picture of a cell, and the brown looking coffee bean things, those are the mitochondria, right? So right here, these are your mitochondria. And these are in the cytosol of your cell. And so if I zoom in on a mitochondria, you see there's a lot going on here. I want to just point out a couple of things. There's two membranes. We have an outer membrane, okay? That's the outermost membrane. Then we have an inner membrane. So you get inside the outer membrane, but to really get inside the mitochondria, you have to cross through another membrane, okay? Now between these two membranes, we have the intermembrane space. So remember that word for later, especially for tomorrow, the intermembrane space. This is actually where ATP is going to be produced, but the process that gets ready to make the ATP all happens within the inner membrane, and we call that the matrix, the space inside both membranes. Once you get all the way to the middle, that's the matrix, okay? Now the inner membrane looks really crazy. It's got all these folds in them that we call cristae, but within those folds, within the inner membrane is the matrix, and that is where oxidative phosphorylation and the Krebs cycle, citric acid cycle happens, okay? And that's what this figure is showing us, okay? I thought this little zoom in was cool too. If you zoom in on the fold of the inner membrane, so this is the inner membrane right here. All these, they're trying to portray to you all these little stick looking things is ATP synthase. It's an enzyme, it's a protein. This is the turbine protein that I said is going to rotate and produce ATP for us, okay? So those are sticking out of the fold of the inner membrane, and that's where the ATP production happens. So this graphic is really helpful for that too. If we're in the matrix, this is where the citric acid cycle happens. Citric acid cycle directly feeds oxidative phosphorylation, and the crossover of all these hydrogen ions across the membrane, turning that protein, is what's going to make ATP. And this actually is in the inner membrane space, okay? All right, so that helps you get oriented a little bit. But mitochondria, that's what makes all the energy to power metabolism. Now let's actually define metabolism, because metabolism is quite complex if you start to look at the details, but a kind of a blanket summary definition is just that it's the sum of all chemical reactions that take place in an organism to maintain life. How many people have heard someone say, I just have a really slow metabolism. My metabolism is just slow. As you get older, your metabolism will slow down. Your metabolism is not slow. Chemical reactions are not slow, okay? Now, everybody has things that affects their metabolism, yes. Age does, your actual physical environment does, your stress levels, the amount of physical activity that you do, all of that affects your metabolism. But it's kind of silly that we say that, because our metabolism is just our chemical reactions that are taking place. It's not, I don't know. Sometimes it sounds like something different when we say it's really slow, but that is metabolism. Now, the way metabolism works is it's got kind of two facets to it. So overall, we have the process of catabolism. So metabolism can either take large molecules and break them down into smaller molecules. That is catabolism. All right, we have to take the food we eat and break it down into metabolites. Those metabolites are useful for other things. And then sometimes, and that, by the way, releases energy. All right, catabolism releases energy in the form of ATP. It helps maintain body temperature. But then if we need to build things, like sometimes the body needs to take the small metabolites that we use from the things we break down, and it needs to synthesize its own stuff. All right, maybe it needs to make a protein, or maybe it needs to make a larger molecule. And so we can take those smaller molecules. It requires energy when we do this. And this is called anabolism, when we build larger molecules with the smaller things we've broken down. Okay, and this is just a cycle. It just feeds itself, okay? All right, so both of those things are used. And now we're gonna kind of transition into how, we're gonna start to look at some specific strategies for how metabolism functions and how it works. What does the body do? It's intricate, it's really beautiful, how this all feeds together and works together. So two really here. Pathways and coupled reactions are the two major strategies that the body uses. So if I ask you that on exam, what are the strategies that the body uses? Describe those to me, or list them, or whatever. Pathways and coupled reactions, okay? So first, what is a metabolic pathway? Well, it's a sequence of reactions. We have been really, really used to in this class looking at an individual reaction. Here's your reactant, what product do you get? Or here's your reactant, here's your product. Tell me something about what kind of reaction is this? You're used to looking at one reaction. Now we're going to have to get used to looking at a chain of reactions. And that is what is called a sequence here. A sequence is like a chain of reactions. A sequence of reactions in which the product of one sequence is the reactant for the next sequence of reactions, okay? So we briefly introduced that yesterday when we talked about feedback inhibition, where we have reactant 1, enzyme 1 makes product B, and product B was turned into C by enzyme 2, and so on. That was like a pathway, a small pathway, okay? But now metabolism, if you look at this picture, is like 4 different pathways built on top of each other, okay? So this is kind of, I love this figure because it's kind of the whole grand scheme of metabolism. It's not everything, but it's a pretty good summary. So I'm just going to introduce this to you in 4 stages, okay? And I definitely want you to know the order of these things, if I had you order them on the exam. Starting out, stage 1 is digestion. We obviously eat food. Your body, when you eat food, it doesn't see good food and bad food. It doesn't see, oh, that food was really good for me. That one wasn't so good for me, the way we think about food. Your body just sees macronutrients. It either sees that you put in lipids, overall you put in carbohydrates, or overall you put in proteins. Okay, every single food, the nutrient that is the largest portion of that food is the macronutrient for that food. So if you eat a burger, protein, okay? If you eat a piece of bread or some crackers, the macronutrient would be carbohydrates, okay? If you eat an avocado, or I don't know if avocado is the best example, but foods high in fat, the major macronutrient there is the lipid, all right? So it takes lipids, carbohydrates, and proteins, and it begins to break them down. And that is stage 1, that's digestion. Lipids become fatty acids and glycerol. Carbohydrates become glucose. Other sugars too. Proteins are broken down into amino acids. That is digestion. Now we're going to really look into the breakdown of lipids and carbs next week. Okay, we're going to talk about glycolysis and beta oxidation next week. But once we break these things down, the major thing, all of these pathways you can see lead to these 2 molecules here. Pyruvate is the end molecule of glycolysis, the final product of glycolysis. Now, don't worry, I'm not going to have you draw the structure of pyruvate. We'll get to that next week. But pyruvate and then these other pathways feed into the production of a molecule called acetyl-CoA, right? So that is where we hit stage 2. Once we've produced acetyl-CoA, now we're in stage 2 of metabolism, okay? So what is acetyl-CoA? Well, you guys might not remember, but an acetyl group, where it gets the name from, is a carbonyl carbon, or it's a carbonyl group, I should say, with a methyl group branched off to the side. That is an acetyl group. Everything in pink here, that's an acetyl group. Now, it is bonded to an enzyme or a coenzyme called coenzyme A. All right, so the combination of this acetyl group and coenzyme A is the molecule we're talking about, acetyl-CoA. All right, so you might see this abbreviated CoA. That is, co stands for coenzyme. And we know what coenzymes do now, right? They aid enzymes. They bind to and they help enzymes function. All right. So this molecule is really, really, really important, acetyl-CoA. Why? Because it specifically, it's like the reactant molecule. It feeds directly into the Krebs cycle. Okay. So at the end of stage 2, when we've made acetyl-CoA, we then feed into the Krebs cycle, or the citric acid cycle. You'll hear me say both. That is stage 3. Okay, we're going to get to stage 3 and stage 4 tomorrow. But once the citric acid cycle runs, what we produce from the citric acid cycle, we do produce some carbon dioxide and 1 molecule of ATP, but the major thing that we produce from the citric acid cycle is this thing right here called reduced coenzymes. And we're going to look at those specifically in the slides later today. Okay, once we get reduced coenzymes, those feed directly into the electron transport chain, which is the final stage, because the electron transport chain, the end product of that is ATP, okay? So you see how this is a giant pathway where the product of one thing feeds into the next thing, and the product of that thing becomes the reactant for the next pathway, okay? So that's why we call it a sequence. All right, any questions so far? Okay. Now, I want to shift gears and mention ATP for just a second and talk about the energy transfer associated with ATP. Then we're going to get into coupled reactions. So let's look at this molecule of ATP for a minute. Adenosine triphosphate. I want you guys to be familiar. I've introduced phosphate groups before, but I'll be honest, we have not spent a ton of time talking about phosphate groups. Okay, so phosphate groups, PO4 with a -3 charge, all right, when it's by itself individually. That is a phosphate group. And what you see is that these phosphate groups are linked together in ATP. Remember that when they're, depending on how many there are, we use prefixes. Here it's triphosphate. But ATP is going to cycle back and forth between its triphosphate form and diphosphate form. Okay, we'll see that in the figure below. So you might see ATP or ADP, and the T stands for tri and the D stands for di, indicating that there's either 3 or 2 phosphate groups attached to ATP. Now, the bond that's really, really important here, these right here, I'm going to highlight them. If you, I don't know if you can y'all see that yellow? Okay, this is an ester bond, but it's a phosphoester bond. So we know what an ester is. Let me draw an ester to remind you. An ester is this: when we have a carbon double bonded to an oxygen, we have a carbonyl group, and we have an oxygen and then some other carbon group. That is what we were introduced as an ester. We're calling this in ATP a phosphoester. And I'm going to finish highlighting the rest of the group, a phosphoester. So instead of carbon, we just have a phosphorus. Okay, so that's why we call it a phosphoester. So if I say it's the phosphoester bond that breaks, or if I ask you what bond in ATP breaks to yield a lot of energy, you would need to tell me the phosphoester bond. Okay, that is the bond that's broken in hydrolysis to yield ADP. That's what releases energy. Well, how much energy? Okay, it's known as the energy currency molecule. How much energy are we talking? I mean, in my head, when people really amp up ATP, I think, oh, lots and lots and lots of energy, like the most energy ever. Well, that's not actually the case. Okay, I'm going to fill in the blank over here. The free energy of ATP hydrolysis is actually an intermediate value. Okay, and this is very, very favorable and actually preferred by the body. Okay, so the ATP or the hydrolysis of ATP doesn't release just gobs and gobs of energy. It releases some energy. And why is that the case? Why is that preferable? Okay, well, look at this table. I'm going to prove to you that it's an intermediate value. This has a bunch of different phosphate molecules. So some kind of molecule that has a phosphate group on it, when you hydrolyze it with water, we yield the molecule in its alcohol form plus some kind of, we call this an inorganic phosphate group, but some kind of phosphate group, okay? This is an important reaction. And you can see a bunch of molecules here. You don't have to know the structure of all these, but a bunch of these have phosphate, okay, in the name somewhere. So there's some kind of phosphate group attached to these molecules. And look at the associated delta G values. Okay, so the hydrolysis of phosphoenol pyruvate yields, wow, negative 14.8 kcal. That's really, really energetically favorable. It's just the hydrolysis of one molecule, okay? But look at ATP. As you go down the table, you decrease in number. ATP is only about half that. So why is ATP the molecule that we use then? Why don't we hydrolyze? Why isn't phosphoenol pyruvate the molecule that we use to power things? It would release a lot more energy. Can anybody think of why we wouldn't want to use that molecule? Everything in life has a cost-benefit analysis. What's the cost-benefit analysis here?
Is it because it has an alcohol group on it?
Not because it has an alcohol group. Look at the energy term. Our body has to make ATP, right? That's where this figure is going to come into play. This figure may help us answer this question. So it is very favorable when we hydrolyze ATP. It releases energy. We use that energy to go do other stuff. But we have to make ATP. So if the hydrolysis of ATP yielded just a very high amount of energy, it would be extremely costly for the body to make ATP. Does that make sense? The more energy it releases, the more energy we have to pay to make it. Okay, anybody like heard about the housing market in Conway right now? Oh, you could sell your house and make a ton of money. No, nobody's keeping up with the housing markets. Just me. This is probably a silly example though, but right now, if anybody were to put their house up for sale, you would get like above your asking price. Okay, and that seems like cool, we should sell our house and make a lot of money. But if it's that way for everybody, you're just going to lose all that money overpaying for the next house you buy. Does that make sense? Okay, same idea here with ATP. The hydrolysis of ATP is very favorable. It releases a lot of money. We gain a lot from it. But then we have to make ATP. So we're just going to lose all that energy by remaking ATP. Okay, does that make sense? So that's why we don't want ATP to release too much energy, because then it's too costly for us to even make it in the first place for the body. And that's what this figure is showing you here, how this thing cycles again and again and again. So start right here with the hydrolysis of ATP. ATP plus water, we break apart the phosphoester bond. and we get ADP, and then this PI right here, this is what is alluding to, this PI is this little phosphate group that is now broken off of ATP. Okay, and that's how we write it. So PI stands for, you can write it different ways. You could draw it out like this, or you could write PO4. Sorry, you would have to write HPO4 because there's a hydrogen that's bonded to it from water, and then with a -2 charge. Okay, this what I wrote up here in the corner is the same thing essentially, except for with the loss of that hydrogen. If you lose that hydrogen too, you now have a -3 charge. So, but this right here, if I ask you on the exam, like to write the hydrolysis reaction for ATP, you would want to write HPO4 with a -2 charge. Okay, that's the phosphate group that's yielded when you hydrolyze it. Okay, with the hydrogen -2 charge. Okay, so when we hydrolyze ATP, all right, here's the 3 phosphate groups, here's the adenosine. When we hydrolyze it, we release a bunch of energy. How much? -7.3 kcal. That number is important. Know that number, -7.3 kcal per mole. And then we get our little old phosphate group released over here. That's the HPO4 -2. And what's left over is ADP. Okay, so now we've gone from ATP, we've done hydrolysis, now we have ADP. Okay, now the body takes ADP and we put a phosphate group back on it. And that costs us money, or that costs us energy. So when we take a phosphate group and we combine it with ATP, we call this phosphorylation. That is what our ATP synthase enzyme does. And this requires an input of energy into that reaction to yield our product ATP. And that costs us the same amount of energy that's released when we hydrolyze ATP. Okay. All right. So we will come back to ATP at the end of the day tomorrow, but I wanted to kind of give you the framework for why this molecule is important and why it is so critical that the energy value of ATP is an intermediate value. Okay. All right, now let's go back to pathways and coupled reactions. Specifically, we're going to talk about coupled reactions here. So I mentioned this, that we have reactions that have a negative delta G in the body. Those are favorable. We have reactions that have a positive delta G in the body. Those are not favorable. An example of some reactions that are not favorable, but we still got to get them done in the body: synthesis of ATP and reduction of coenzymes. Those are 2 that really apply to our topics today. Okay, so how do we do this? If they're not favorable, how does the body go about making this stuff? We've still got to make these compounds and these molecules so that we can use them in other ways. Well, this is where coupled reactions come in. So we can think about pathways and coupled reactions using this energy diagram. So I'm going to zoom in on this energy diagram on the right. Draw this with me. Say we have our starting reactants and they have X amount of energy, okay, the highest amount of energy. Overall pathways follow exergonic reactions, okay? So what that means is that every step in the pathway has got to be favorable for the body to do it. Ultimately, at the end of the day, it's got to be favorable. So we can think of these favorable reactions as downward arrows. So the take our reactants, do step number 1, and we get some kind of product. We'll call this product A. Okay, that's got to be favorable. It's got to release energy. A is now lower in energy than our starting reactants. Okay, then maybe we have step number 2, and we make product B. Okay, then to get our final products, maybe we have this big long step down at the bottom, step number 3, and we get our final products. Okay, so you think of these as downward reactions, and they're releasing energy all the way. Well, what if we have a reaction that's not favorable? We would think of that as like an upward arrow, okay, or an uphill reaction. And the way we have those happen is we couple it to a larger downhill reaction. The way we make unfavorable reactions happen is we couple them to a larger downhill reaction. So that'd be like this. We have an endergonic reaction that needs to take place. We're going to couple it. Maybe it only requires that much energy. We're going to couple it to a larger downhill reaction. They're going to happen on the same enzyme at the same time, and the energy released from the downward arrow is going to be used to power or fuel the up arrow. Okay, so this would be an example right here of a coupled reaction. Now, if this is still not making sense in your brain, I want you to look at this little wagon figure. Okay, I know this seems silly, but it will make a lot of sense. I want you to think, look at these figures. We have two wagons. We have one going uphill. That requires energy to walk uphill, right? So we think of that as our positive delta G. The body has to invest energy to make this wagon go uphill or to make an endergonic reaction happen. So this is our endergonic reaction. And the way the body does this is it couples it to, or it ties it to, we have a larger wagon. Notice the size difference. This would be our exergonic or a favorable spontaneous reaction. It couples it to a larger downhill reaction with a negative delta G. Okay, so hopefully that figure kind of helps explain what the energy diagram was trying to explain. The way you will see this written out in your textbook and the way you'll see it on the exam is like this. Let's say we have some kind of unfavorable reaction, all right? So we're gonna phosphorylate glucose. This is, you actually should be familiar with this reaction. I mentioned it a couple of times. I mentioned it yesterday. Remember G6P yesterday when we were talking about inhibition? So the phosphorylation of glucose to make G6P, oh, I'm gonna highlight it. This is G6P. This is the first step in glycolysis. This is actually not a favorable reaction. So how the heck do we make it happen if it's not favorable? Okay, it requires an investment of 3.3 kcal of energy. What we do is we couple it to a favorable reaction. We know the hydrolysis of ATP is favorable. It releases, I should highlight, it releases 7.3 kcal of energy. So that's why the sign is negative. Negative is favorable, positive is not favorable. Positive costs the body energy, negative releases energy for the body to use. So we just couple these things. And the way you need to write this out, you're gonna have to do this on the exam, we're gonna do a practice problem in a minute, is we look at both sides of these two equations and we cross out anything that's on both sides. So do y'all see anything between these top two reactions here, the favorable and the unfavorable one? Do y'all see anything that's on both sides of the arrows?
The H2O.
Do what?
The H2O.
H2O, let's cross that out, okay? So you have to write out both reactions fully, and then you assess, do I have anything I can cross out on both sides? And then you write the final reaction, okay? So the final reaction, we've got glucose, ATP. Oh, we also have a phosphate group on both sides. We can cross that out. So what we have overall is glucose plus ATP makes glucose 6-phosphate and ADP, okay? And you have this hydrogen ion too. They didn't write that in, plus H plus. All right, and then we add up the delta G's and we see, okay, overall, once we couple these reactions, now, even though we had to pay 3.3 moles of energy, we still overall release -4 kcals. And that is how we make endergonic reactions happen, by coupling with the larger exergonic reactions. Does that make sense? So coupled reactions, this strategy in metabolism allows non-spontaneous reactions to occur in the body.
On the test, would you have it written out just like that, unfavorable, favorable, and then the reaction, and all we would have to do is mark out and write the end product?
Well, Robin, we're actually doing some practice problems, and it could look like any of these practice problems we're about to do.
Okay.
So I have a poll question. I want you to look at this question right here while I launch the poll. Based on what we've talked about. Oh, I didn't title this one. Based on what we talked about, what do you think? How is ATP hydrolysis used to power other cellular reactions? All right, two more. Last one. All right, in Paul. Okay, I'm glad we're talking about this. So we got some different answers here. Most people said, so the question is, how is ATP hydrolysis used to power other cellular reactions? Most people said A, it is coupled with endergonic reactions, okay? Nobody picked B, produce water to fuel other reactions, okay? B is not correct. So somebody said C, it adds a phosphate group to ADP. So the hydrolysis of ATP, even just the inherent, if you read this, the hydrolysis of ATP just breaks apart a phosphate from ATP and it yields ADP in the process. So phosphorylation, this is incorrect because what this is describing is phosphorylation. Of ATP, I should say ADP, not hydrolysis of ATP, okay? This, in phosphorylation of ADP, we do add a phosphate group to ADP, but then we make ATP. We're asking about the opposite process, hydrolysis of ATP. So that's why C is not correct. And this is not how we power other cellular reactions, by phosphorylating ADP. And enzymatic catalyst, ATP is not an enzyme, okay? It's just a molecule. It's not a protein, okay? So if it's not a protein and it's not an enzyme, it can't have any catalytic power or activity. So D is not correct either. So it has to be A. It is coupled, and that's what we were trying to show right here, is how we couple these reactions together to yield overall a spontaneous favorable reaction, okay? Coupled reactions, that's the key. One of the main metabolic strategies, okay? Let's do another practice problem with this. All right, it says the hydrolysis of succinyl-CoA is coupled with production of GTP, which looks a whole lot like ATP, okay? It's kind of like the cousin to ATP. The equations for the reaction are given below. Combine the equations appropriately and determine if the coupled reaction is favorable. So these two reactions are written out for you. Robin, this is kind of what you were asking. Oh, I need to stop sharing the call. I want you to write the end product. So if you need to, draw a line right here, cross out anything on both sides, add your delta G's together, and write your final equation, and then tell me, is it favorable or not favorable? I want you to go ahead and try to do that without me, and then I'm going to do it.
Sorry, did you add that slide? Because I don't have it.
Yeah, this is the one I mentioned this at the beginning. I added this in last night. You have this question. Do you have this question? That first question? Yeah, I just added the first one on there. So take a screenshot if you need to, or write these problems down. Succinyl-CoA makes succinate and CoA. Your delta G is negative 9.4 kcal. Sorry about that. It was just late when I added this. I just realized we really needed to do more practice on this. And then you got GDP in the second reaction plus a phosphate group. It just they wrote this phosphate group a little bit different. You'll normally see me write it like HPO4 with a -2 charge. Same thing here, same number. They just wrote HOPO3, but that's the same thing as HPO4 with -2 charge. Okay, plus hydrogen plus GTP, and your delta G there. You don't have to write the kilojoule numbers. Okay, just focus on the kcal per mole. If you can cross anything out, do it, and then add up your delta G's. So I'm going to go ahead and write the final reaction too. You should have succinyl-CoA plus GDP plus the phosphate group. And I don't think we can cross anything out. So we get succinate. Oh, I forgot, plus the hydrogen here. We get succinate plus CoA plus GTP plus H2O. Okay, so it's a long reaction. What's your delta G overall? What did you guys get?
I got negative 2.1.
Y'all agree with Madison? kcal per mole. Don't forget the units. So if you want to have a calculator for Monday, this is the only kind of math you're gonna have to do. It'll be simple numbers like this. So if you feel comfortable without a calculator, but bring one in case you want one. So is this favorable or not favorable when you couple these reactions together?
Favorable because it's a negative number.
Exactly right, favorable. And I mean, look at this. It cost us a lot of energy, 7.3 kcal per mole to make GDP or to phosphorylate GDP. But we can do it when we couple it with a larger negative delta G reaction, okay? All right. This one, I don't think you have either. Again, sorry for this. I should have emailed these to you guys. I can email these real quick if y'all want that. I didn't think to do that this morning. So read this problem. I'm going to email y'all the PDF that has the updated slides. Yeah, this problem should be just like the one before, but you actually have to write it out this time. There it is. Okay, I'm sending them now. So if you don't get all this written down, you'll have this slide emailed to you now. There might be a lot of extra slides on the end of that PDF that we're not getting to, so ignore the bare base slides at the end. Hydrolysis of acetyl phosphate to give acetate and hydrogen phosphate ion has a delta G of negative 10.3 kcal per mole. So let's try to write this out. And you can use abbreviations, all right? Maybe for you, you're gonna write out the whole word this one. Acetyl phosphate, okay? So we have this acetyl group. Remember, this is an acetyl group, and we have some sort of phosphate. We got negative charges right there. So that's acetyl phosphate to give acetate and hydrogen phosphate. So acetate would be this. So we're gonna break the bond here. Now we're gonna break this phosphodiester bond. So acetate, which bond are we gonna break? Right there. And we're gonna use water. So I forgot a reactant. You gotta have water. It says hydrolysis of this. So we've got to use water. Anytime we're doing hydrolysis, water is a reactant. All right. Actually, I think the bond, now that I remember we've got water, it's one bond over. We're gonna break this bond. Either way, you should be able to still write the same products, okay? So acetate, the acetate anion. There's acetate. And then a hydrogen phosphate is just the phosphate group. You could write it like this, HOPO3 with a -2, or you can write HPO4 -2. And this delta G is a negative 10.3 kcal per mole. All right, so there's hydrolysis of acetyl phosphate to give acetate and hydrogen phosphate. And then we have combine the equation and delta G values to determine whether coupling of this reaction with phosphorylation of ADP to produce ATP is favorable. Okay, so we can write that reaction out. We're gonna phosphorylate ADP. So we got ADP plus we got a phosphate group, HOPO3 minus 2. Or again, you can write HPO4 minus 2. Same thing. And we're gonna get ATP. And water. All right, and the delta G there, anybody remember the value associated with the delta G? How much energy does it cost us to make? You can go look at this figure right up here if you need to. And by the way, if you're wondering, how did she get this reaction? What I just wrote is this in reverse, except for we left off this hydrogen, didn't we? And it would be this number in reverse if we're doing the opposite. If we're taking our products and then making our reactants, we got to write a positive 7.3 kcal per mole. So we have the hydrogen there. Positive 7.3 kcal per mole. Okay, so now what we're asked to do is cross off anything on both sides, add up your delta G's, and get the coupled reaction with the delta G for the coupled reaction. So I can cross off water. I can cross off this phosphate group. I think that's all I can cross off. And I'm going to abbreviate for time's sake. This is acetyl. I'm going to write ACETYLP. Were some of you, anybody concerned with drawing that? Like that would be a challenge for you on the exam. Like how did you know how to draw that?
Yes, I would rather
Me.
not.
Okay. I'm not going to ask you to draw something you've never drawn before. I recognize we've never drawn this, okay? I'm not going to ask you to draw something that you've never, ever, ever drawn before unless I know that you know that I know you have the knowledge to do it. Like for example, on the exam, I've given you reactions where I say, here's the reactants, draw the product. Some of these products you've never drawn before, but you should have the knowledge inherent to draw that, okay? I'm not going to try to pick something ultimately deceptive. Does that make sense? I see some worried looks.
You could just put the equation for us.
I might give you the reactant molecule and then say, draw the product, okay? But I've been doing that the past 2 weeks. So that's stuff y'all should feel comfortable with.
My brain hurts.
I'm not
No.
comfortable with the class.
Do what, Cameron?
I said I'm not comfortable with the class.
Not comfortable. Well, maybe I should choose my words more wisely. All right, we got acetyl phosphate, plus we got some sort of hydrogen, and we have ADP. Those are the reactants that we were not able to cross any of these out on the reactant side. Oops. Okay, so that's why we wrote all these. I think that's all of them. Now on the product side, you write everything that you didn't cross out. So we got acetyl here. I'm just going to write acetyl, or sorry, acetate. That's not acetyl, that's acetate. Whoops. All right, acetate plus ATP. And I think that's our only products. What is our delta G overall? We should get -3, right? Or if I give you the typed out reactions, you should be able to write the product reaction and add up your delta G. Are y'all at least good with that?
So you're pretty much saying we're gonna have to draw some.
Always.
Oh.
I mean, here's the deal. I could give y'all something like this and say, you know, write write the product equation and add up your delta G, and then tell me something about it based on what you get. Okay.
I liked that.
Okay. Good. I'm glad you like that. All right. So that's some practice. I want you to know coupled reactions, why they're important. Recognize the examples that we've used. ATP is all up in this because that is how ATP is used in the body. All right, another metabolic strategy though, in addition to pathways and coupled reactions, is oxidize and reduce coenzymes. Okay, so let's talk about those. And this is our last slide for the day. Okay. So we're going to be here for just a minute. Now, oxidation reduction. Let's remind ourselves of a few things. I want to fill in these blanks. Oxidation is, can anybody fill these in for me?
Is it the loss of electrons?
Yes, Abby, loss of electrons or hydrogen. So if we're removing hydrogens off of a molecule, we're oxidizing it. Or it can be the gain of oxygen. Okay, so we can oxidize something by increasing the number of carbon-oxygen bonds, decreasing the number of carbon-hydrogen bonds, or removing electrons. Reduction is just the opposite. It is the gain of electrons or hydrogen. So you're making carbon-hydrogen bonds, or the loss of oxygen. Oxidation reduction always occur together. These are also coupled reactions, okay? So I mentioned this earlier, that oxidation reactions, I'm gonna put a star here, these are favorable. It just so happens that they usually always have a negative delta G if you look at the energy value associated with oxidation reactions, right? But reduction reactions have to occur as well, okay? And metabolism, when you really begin to look at it close up and then zoom back out and look at it big picture, you see, oh, it's just honestly a chain of redox reactions. Oxidation reduction, oxidation reaction. We're just passing along electrons or hydrogens or energy in some form until we get our final product. So if this is gonna run, if this whole train is gonna run, we've got to have some wheels for it to run on, okay? And so we've got to have a steady supply of oxidizing and reducing agents. If we're going to do a redox reaction, we've got to have a supply of an oxidizing or reducing agent, something to transport the electrons. And that is where coenzymes come into play. So I want to look at this figure because these coenzymes are key to metabolism, all right, all throughout the pathways. You can cycle them back and forth. What's nice about them is they just cycle back and forth between their oxidized and reduced states. Okay, they have a really boring life. They just show up to work, they get oxidized, they get reduced, back and forth, and they go home, okay? So let's look at molecule A. Molecule A has 2 hydrogens, all right? We're gonna take these hydrogens off of molecule A. So are we reducing, or are we oxidizing molecule A here? Let's go from this to this. Is this oxidation or reduction?
Oxidation.
Yep. So we're going to oxidize this molecule. So we've got to have something else that's going to be reduced in the process. And that's where the coenzyme comes into play. The coenzyme starts out in its oxidized form. It receives these hydrogens and it's reduced in the process. And by it being reduced, molecule A is oxidized. Okay. Now let's take molecule B. We got a molecule in our pathway somewhere that we're going to put hydrogens on. Are we oxidizing or reducing molecule B? Reducing it, right? So for this to be reduced, something has to be oxidized. So we're going to take the coenzyme that we reduced earlier, pass those hydrogens on. In the process of reducing molecule B, the coenzyme will be oxidized. Okay, you see how they always work in tandem. Okay, and the cycle just goes on and on. It might not always be the same 2 molecule A and B. It might be different molecules, but throughout the pathway, this just happens again and again and again. Okay, so let's look at a real example of a real coenzyme. We introduced this yesterday when we were talking about vitamins and minerals. We introduced NAD as a cofactor to coenzyme niacin. We introduced niacin as a cofactor to this coenzyme NAD. All right, that was on yesterday's slides at the very end. So here's the molecule. You don't need to like memorize this whole molecule. But I do want to show you that, oh, by the way, real quick, so you can have NAD or NADP. So what's the difference between the structures here? Why do we have two different ones? The only thing that's different is that in NADPH, this OH group is a phosphate group, okay? And that's NADP, right? So in this structure, I want you to look right up here. Right now, NAD plus is the oxidized form, right? And we have this carbon that has 4 bonds, 1 to hydrogen. When we reduce this, the difference here, same ring that I've boxed in over there, we add another hydrogen to that carbon and the double bond goes away. Some electrons are moved around, the double bond goes away. This carbon still has 4 bonds, but now 2 of them are to hydrogen. So we reduce that carbon, right? Because we said reduction is the gain of electrons or hydrogen. So we added a hydrogen on here, we reduced NAD. Now we call it NADH. The H indicates the added hydrogen that we've just put on. And this is the reduced form. Okay. So this is an example of a real biological molecule that is used in our body every single day. Reduction and oxidation, and this is going to power metabolism. It's going to allow metabolism to happen. Now, so let me read these statements. As an oxidizing agent, NAD plus and NADP remove hydrogen from a substrate. If they're removing hydrogen from a substrate, they are taking the hydrogen and putting it on themselves. As a reducing agent, NADH and NADPH add hydrogen to a substrate. So it would be this in reverse. We would just turn this arrow around and say we've got the reduced form. If we're going to reduce a molecule, we're going to add a hydrogen to that molecule, but we ourselves are losing a hydrogen. So we're going to be oxidized if we're the coenzyme. Okay. Same thing can happen with FAD. FAD is the cousin to NAD. They don't look the same, but they do the same thing. It's another coenzyme. I encourage you to please go look at page 674 before the exam on Monday, and then familiarize with the same reduced and oxidized forms of FAD and FADH. If I were to ask you or give you pictures and say which one's the oxidized or the reduced forms, I would want you to be able to tell me, okay? This is how it can look in a real biological reaction. So this is how we represent this in like the citric acid cycle. You'll see this looped arrow. So we've got malate, which is an intermediate in the citric acid cycle. And what happens is that malate has to be oxidized, okay, to oxaloacetate. And we do that with coenzyme NAD+, okay? So it's in its oxidized form. It's going to grab, look, in pink, these hydrogens, I don't know if y'all can see this, I'm going to go to black. It's going to grab the hydrogen here and the hydrogen here off malate, and then you form a double bond right here on this carbon. That's a very small, seemingly technical change, but that in itself is one step in the citric acid cycle. In the process, NAD+ is reduced so that malate can be oxidized, okay? And so you'll see it written like this with the curved arrow. So this is a redox reaction. The coenzyme is being reduced, the substrate malate is being oxidized. Okay. So any questions on oxidation reaction or oxidation reduction? We're actually kind of getting done early today. That's probably kind of nice. We haven't had a day like this so far. But I wanted to take our time and really walk through these things. I could probably shove 15 slides into an hour and a half, but that would just be overwhelming. We've missed so many details. Nobody would learn
I have a
anything.
question.
Yes, Maggie.
I'm kind of confused on the figure that's right next to the one we just looked at, or right above.
Right here, the picture of the molecule NAD.
Yeah. Why does it? Can you explain why it says this OH group is a phosphate group?
So we have two possible forms of NAD. We have NAD and we have NADP. So I was just telling you what the P stands for. Okay, the NADP is the same exact molecule just instead of an OH, we would have like this. Instead of an OH here, let me show you what we have. We'd have a phosphate group. So it would look like this, O, P, like that, okay? Does that make sense? This phosphate group might have, that charge doesn't make sense, it might have hydrogens here. So that overall you still get this plus charge. But regardless, you have a phosphate group there instead of an OH group. That's the structural difference between NAD and NADP. They are still oxidized, they still cycle back between their reduced and their oxidized forms the same way. The site of reduction is the same. This carbon right here gets a bond to a hydrogen that's reduced. And so in real chemical reactions, when we cycle those through, we see the addition of hydrogens to our coenzyme and the loss of hydrogens from the substrate, so that the substrate is oxidized, okay?
Can you show me again how you knew, like, other than it saying that that's the site of reduction, how would you know that that was where you take it from? Does that make sense?
Oh, people have just, and you have to do experiments on this in the lab. So the amazing part is, how do we learn all this? Literally, people isolate these molecules in the lab. They weigh the size of them, and they use instruments to determine the size and what piece of the molecule is the same. It's really, really complex, Abby, how we have actually learned this. What I'm showing you in the slide, telling you this figure has probably taken tons and tons and tons of research.
So you're not saying we'd have that reaction and then have to show where it came from? You were just showing us. Does that make sense?
So if I showed you a picture of this molecule and said, is this the oxidized or the reduced form of NAD? Could you tell me?
Oh, okay, I see. We would look at the reduced and see which molecule.
You'd say, okay, is there one hydrogen here or is there two hydrogens here?
Oh, okay.
But you need to know which part of the molecule to look at, which is why I'm pointing it out.
Okay, thank you.
Because there's lots of other hydrogens on the molecule, right? Lots of other carbons, but the one that matters is the one that I've got highlighted. Okay. Y'all feel okay with the redox stuff?
Yes.
Okay. What about writing out the coupled reactions?
Boo.
What about this idea with the pathways and looking at the energy diagram? Did that make sense of explaining how coupled reactions work?
Woohoo. That one was good.
Okay, which one? The wagon?
The woohoo, the couple reaction.
Okay, good. All right, so I'm just kind of scrolling back through the slides. If y'all have any questions that you're like, oh, I wanted to ask this and I just didn't, now's the time. This is as far as I want to get today. I want to save Krebs cycle and oxidative phosphorylation for tomorrow. So I kind of really like slowing down through this chapter and taking it in portions. We're gonna try to go. I can't promise everything will be a slower pace till the end, but I'm gonna try to slow down some so that the biochemistry isn't just glazed over.
So this test will only have 3 chapters?
3 chapters.
More room on my note card.
More room on your note card, except for all those amino acid side chains.
Yeah. Oh yeah.
Okay. I definitely want you to know the structure of mitochondria. If I give you a figure or whatever, be able to label it. Tell me where. Electron transport chain, which we will go over tomorrow, but by the end of tomorrow, you should be able to tell me where the Krebs cycle happens, where ATP is produced. I want you to know why delta G is a good indicator for determining if a reaction is favorable or not, okay? If I give you a reaction diagram, could you tell me, oh, that's favorable or not favorable based on what we drew? Do you understand the differences between the exergonic one and the endergonic one? The energy level of the products. and whether your delta G overall is positive or negative.
I know you already explained it, but the reason why delta G is important, would it just be as simple as saying like it tells if it's negative or positive, which tells if it's like exergonic or
Yeah, Claire, that's why it's so nice. Instead of measuring 3 different variables, we're just looking at the sign of 1.
okay.
And it encompasses all those 3 that are important.
Okay. Thanks.
Okay. Well, what we're gonna do then is take a nice, do y'all wanna get started at 10:45 and get rolling, or do you want a longer break than that?
Depends on how long the lab's gonna take.
Well, let's look at the lab really quick. So before we go, I'm gonna share this over. We have, I can just import it. There we go. Right here. So this is the lab. So look, 4 problems. But that doesn't necessarily mean it's easy, right? We only had 3 or 4 yesterday, and that lab was kind of challenging. So just take a look at it. This is what we're going to be doing when we come back. Complete the following reactions, write the products, and then tell me what enzyme was used. So what class of enzymes? So you have 6 major classes of enzymes. You can tell me specifically the subclass or the main class. And that's it. But I think 3 is especially hard. But the others, you should really, really, really look at. Did it do it? Oh, Robin, what happened? Okay, let's try this again. There we go. You should really look at these tables, okay, right here, and look at the examples. The examples will help you understand what's going on some of them, not all of them.